Hi everyone, my dear students. Hope you are doing good for module exam. As you asked about plasma proteins, today my topic is about plasma protein functions with the disorders I have given. So first, in this topic, we'll be saying what is plasma protein, how far it functions, what are the different types of plasma proteins with the disorders, complaints I'll be giving. So very important detailed question for university exam as well as IA exam. Here, plasma proteins greater than 300 different proteins contain, but many pathological conditions affect level of plasma proteins. And here, most of the plasma proteins are synthesized in liver, except immunoglobulin. All the plasma proteins except immunoglobulin synthesized in the liver. And another one thing I want to tell you, all plasma protein except albumin are glycoproteins. So mostly some are produced in other sites I have given here. It may be example of immunoglobulin. So other may be synthesized in the liver. A normal adult contains approximately 70 gram per ml of plasma proteins. So next, functions of plasma protein, you have to concentrate the important thing. For example, albumin are plasma proteins we are telling. So very important thing is transport. So here transport functions may be albumin, pre-albumin and globulins. Maintenance of plasma oncotic pressure. Albumin plays an important role. As I've already told in the membrane transport class, you should differentiate what is meant by oncotic pressure and osmotic pressure. It may be asked in the university exams. So oncotic pressure, intake of water into the circulatory part, into the blood capillaries. Whereas osmotic pressure, it drives the water from lower to higher solute concentrations. Clear? So you should know the difference between oncotic pressure and osmotic pressure. And defense immunoglobulins and other complement systems are involved in defense mechanism. Clotting and fibrinolysis involved by thrombin and plasmin. So these are the important functions of plasma proteins. Transport, maintenance of plasma oncotic pressure, defense mechanism, clotting and fibrinolysis. Measurement of plasma protein plays an important topic here, subtopic, because plasma proteins are identified by electrophoresis technique. So we'll be seeing the small 3 to 4 slides, may be asked in 5-mark question also. So it can be measured by quantitative measurement of a specific protein, chemical or immunological reactions, and semi-quantitative measurement by electrophoresis. Here the proteins are separated by electrical charge in electrophoresis. Staining reagents may be asked in MCQ question, multiple choice question, Kumasi brilliant blue stains, either R250 or G250, and silver stains. 5 separate bands at last you will be getting. It will be observed. These bands may change in the disorders, which will be explained in the later slides. So for 5-mark question, electrophoresis, they'll be asking what is the principle. First, you have to write the definition, principle, procedure, and different patterns of electrophoresis. For conditions you have to write. So electrophoresis, this is the definition. It may be defined as a migration of charged particles through a solution under the influence of an external electrical field. So definition as such, you just mug it up because it's very important. For each and every technique, you should write the definition. So principle of electrophoresis is a process of moving charged molecules in a solution by applying an electric field across a mixture. So mostly proteins will be traveling from negative to positive, right? Cathode to anode. You have to draw this diagram when you are writing for electrophoresis. These are the different 5 bands. I have already told no 5 bands you will be observing. So albumin, alpha globulins, alpha 1, alpha 2 will be there, beta globulins, at last gamma globulins. So electrophoresis, again I have referred migration of charged particles from negative to positive. So here this is the origin site. It will be traveling from here to this side. Why albumin is going fast means as the molecular weight of albumin is very less, it will be moving faster. As such of obese and slim person running in the ground. So next, what is SP procedure? What is a serum protein electrophoresis procedure? First, sample preparation, gel loading, gel running, protein staining, and then quantification. So just as such, whatever I have shown, you can note it down. No need to explain here. So next, separated protein bands can be quantified using densitometer. So here albumin is the albumin, alpha 1, alpha 2, beta, gamma. So nearest point of origin is gamma globulin, which is having high molecular weight. It can't move as such when compared with albumin. It stays here. So first origin, this is the origin site, and the cathode from protein starts running, and here it will be coming. So this is band, you have to drop it. And normal reference range I have given here, note it down because you have to write it down when you're writing the electrophoresis 5-mark question. Albumin, I have given with the gram per DL as well as with the percentage. Alpha 1, alpha 2, beta and gamma globulin. So this is normal pattern you should draw, because when you're comparing with the case conditions, you should compare with the normal. Without drawing the normal, you won't draw case condition. If you are drawing the condition also, we won't give mark. So better draw the normal pattern. And in that I want to tell alpha 1 otherwise called as, because in MCQ they'll be asking what is the alpha 1 globulin commonly called. So better note it down. Alpha 1 antitrypsin, thyroxine binding globulin, transcontin, etc. And then alpha 2 haptoglobin, ceruloplasmin, macroglobulin. And then beta transferrin lipoprotein, beta lipoprotein. And last gamma globulin, you will be knowing gamma globulin or antibodies. So I have told again negative to positive. So normal pattern of plasma protein is very important for electrophoresis. And these are the different patterns I want to just explain here. So in this way you can remember this is gamma and this is albumin, beta, alpha 2, alpha 1. So different patterns I have shown here. Patterns of immune activation, protein loss, here beta cell dysfunction, immunoglobulin production, for everything, nephrotic syndrome, very important thing. I have just shown this, you can refer. In the later slides, I have drawn the slides for conditions also. So now types of plasma proteins, pre-albumin, albumin, alpha-1. I have given the example before itself, alpha-1 antitrypsin, alpha-fetoprotein, alpha-2 globulins in that ceruloplasmin and haptoglobin, beta-globulin, CRP, transferrin, beta-2 microglobulin, gamma globulins. So this is very important slide, introduction slide for plasma proteins. You should know different names of different bands, right? Alpha 1, alpha 2, you'll be telling what is the other name of alpha 1. Alpha 2 I have given here. So first, before entering into the albumin, pre-albumin, you should note it down. Transthyretin, a transporter protein for thyroid hormones. Only I have given the functions. Functions with the what is that? Electrophoretic patterns. This is important more than you can write in the exam. So thyroid hormones, retinol, vitamin A, it migrates faster than albumin in electrophoresis, separated by immunoelectrophoresis, and where low levels found in liver disease, nephrotic syndrome, acute phase inflammatory response, malnutrition. So short half-life is 2 days. When compared to albumin, this is less only. Okay. Albumin, most abundant plasma protein, approximately 40 gram per L in normal adult. It's synthesized in the liver as pre-proalbumin and then secreted as a albumin. Half-life is 20 days. It rapidly decreased in injury, infection and surgery. Functions main thing is oncotic pressure. With the help of this only albumin oncotic pressure is maintained. As I've already told, pressure exerted by plasma protein that pulls water into the circulatory system that maintains the fluid distribution in and outside of the cell. Along with the maintenance of oncotic pressure, it acts as a specific carrier, non-specific carrier of hormones, calcium, free fatty acids, drugs, etc. With the help of pinocytosis, it is hydrolyzed to amino acids. Useful in treatment of liver disease, hemorrhage, shock, and burns. Yeah, in this, I just want to tell albumin act as a conjugating agent. Along with this, amino acids may be carried from one ICF to ECF or ECF to ICF. Next, hypoalbuminemia causes it decreased albumin synthesis, liver cirrhosis, malnutrition. So wherever when hypoalbuminemia occurs, I have given here. If they are asking write any two conditions that causes hypoalbuminemia, you can know this slide. Increased catabolism in infections, excessive loss in bowel, excretion by the kidneys, and severe burns. Effects, it may be due to low oncotic pressure, edema due to low oncotic pressure, reduced transport of drugs and other substance in plasma, it reduced protein bound calcium. So these are the different effects that leads to hypoalbuminemia. And hyper, no clinical conditions are known, but the only cause is dehydration. Next, alpha-1 antitrypsin synthesized by the liver and macrophages, an acute phase protein. Very important 3-mark question or 4-mark question they may ask. What are acute phase proteins? Classify it. In later slides, I'll be telling. It is a protein that inhibits protease. Proteases you'll be knowing proteolytic enzymes, digestive enzyme, trypsin, chymotrypsin. Other proteases may be elastase and thrombin. So infection leads to protease release from bacteria and from other leukocytes. So I have given normal pattern here, albumin level is increased, but whereas antitrypsin deficiency happen. So here this is alpha 1 is not there. So like that, note the graph and then draw the picture. Types of alpha-1 antitrypsin, over 30 types, but the most common is M. allele. It is nothing but a gene frequency M type. Genetic deficiency of alpha-1 antitrypsin. Synthesis of the defective occurs in the liver, but it cannot secrete the protein. Here it accumulates in the liver, that's why deficiency happens in the plasma. Next, clinical consequence of alpha-1 antitrypsin deficiency, mostly neonatal jaundice with evidence of cholestasis. Clear? So cholestasis you will be knowing, blockage of the bile synthesis from liver to the small intestine. You should know the condition and correlate with the markers. Childhood liver cirrhosis, pulmonary emphysema in young adults, and lab diagnosis, lack of alpha-1 globulin band in protein electrophoresis. I've shown in the graph previous slide. No? So the alpha-1 globulin in protein electrophoresis, it is deficient. And these can be measured by radial immunodiffusion, isoelectric focusing, or nephrometry. Just learn the technique. Whatever the technique we are telling, just learn it. Next, this is very important marker, AFP, alpha fetoprotein, a tumor marker. Important question, 2-mark questions or 1 mark they'll be asking in MCQ. Synthesized in the developing embryo and fetus by parenchymal cells of the liver. The level decreases gradually during intrauterine life and it reach adults level at the birth. Functions is unknown, but it may protect fetus from immunologic attack by the mother, which is nothing but a defense mechanism. So elevated AFP levels are associated with neural tube defect. If there is any neurological disorders, anencephaly, decreased maternal AFP levels associated with increased risk of Down syndrome. Down syndrome. Down syndrome. AFP is a tumor marker for hepatoma and testicular cancer. So ceruloplasmin synthesized by the liver, it contains greater than 90% of serum copper and oxidoreductase that inactivates ROS, reactive oxygen species, causing tissue damage in acute phase response, and very important for iron absorption from the intestine. So it may cause Wilson disease. Due to low plasma levels of ceruloplasmin, copper is accumulated in the liver and brain. So this slide is enough. Ceruloplasmin will be asking the important 2-mark question, and very important thing will be asking Wilson disease. So when compared to ceruloplasmin, Wilson disease, when you are learning about minerals, you'll be taught this. Haptoglobin synthesized by the liver, it binds to free hemoglobin to form complex that are metabolized in reticular endothelial system, limits iron losses by preventing hemoglobin loss from kidneys. So plasma level decreases during hemolysis. Next, transferrin, you'll be knowing iron transport protein. Plasma level drops in malnutrition, liver disease, inflammation, malignancy. And the deficiency results in increased hepatic synthesis, a negative acute phase protein. So transferrin is a negative acute phase protein. Next, beta-2 microglobulin. It is a human leukocyte antigen. It present on the surface of lymphocytes and most nucleated cells, filtered by glomeruli. And elevated serum levels found in overproduction in disease may be a tumor marker for leukemia, lymphomas, multiple myeloma. And this is very important inflammatory marker. They'll be asking for 2 mark question. Acute phase protein synthesized by the liver, important for phagocytosis, engulfing of bacteria or any waste products. High plasma levels found in many inflammatory conditions such as rheumatoid arthritis. So for rheumatoid arthritis, it can be used as a marker, and very important ischemic heart disease marker is CRP. Last, hypergammaglobulinemia. It may result from stimulation of B cells. Polyclonal hypergammaglobulinemia, monoclonal proliferation, paraproteinemia. So these are the terms, very important terms we'll be expecting in the answer sheet. They are asking multiple myeloma. This slide is very important. Polyclonal hypergammaglobulinemia, stimulation of many clones, globulin band appears large in electrophoresis, and clinical conditions, acute and chronic infections, autoimmune disorders, chronic liver disorders. So this is normal pattern and I have given here. Cirrhosis null, at once we have to remember fusion of gamma, last gamma and beta. Fusion happens. So there is no single peak has seen here. Fusion of alpha and beta tells about cirrhosis. Monoclonal hypergammaglobulinemia, proliferation of a single B cell clone produces single type of immunoglobulin. It appears as a separate dense band, paraprotein or M band, which is nothing but multiple myeloma. So paraproteinemia, the paraproteins are very important characteristic features of malignant B cell proliferation. So this is M band. The shape looks like a M band, alphabet M. Clear? So this is albumin and this is gamma globulin and this is normal pattern. I have already told. Each and everything you have to compare with normal pattern with a case. Two different phase proteins, very important 5-mark questions. Acute phase proteins are positive and negative. So plasma protein levels increase in infection, inflammation, malignancy, trauma and surgery. These proteins are called acute short-term phase reactants. It is synthesized due to body's response to injury. Example, very important, alpha-1 antitrypsin, haptoglobin, ceruloplasmin, fibrinogen, C-reactive protein. And these mediators cause these proteins to increase after injury. Cytokines, interleukin 1, interleukin 6, tumor necrosis. And these factors affecting acute phase proteins, interferons, platelet activating factors. So these are the mediators that may increase these proteins. Functions, it bind to polysaccharides in bacterial walls, activate complement system. Complement system is nothing but antigen, antibody clotting factors that will come, stimulate phagocytosis. So I have inflammation. I have shown everything is high, peak in the peak, right? When compared to this. Negative acute phase proteins, these proteins decrease in inflammation. Albumin, prealbumin, transferrin. So very good proteins, they are decreased in inflammation. It mediated by inflammatory response via cytokines and hormones. Synthesis of these proteins decrease to save amino acids for positive acute phase proteins. So that's all my dear ones. I have given the slide for plasma proteins. Very important detailed question or 5-mark question they will ask in the university exam. It is very important. Questions, possible questions. What are plasma proteins? How are they classified? Other thing is acute phase proteins. How they are classified, positive and negative. In that individual they can ask CRP, transferrin, like that they may ask individually. So very important question is plasma protein. Read and kindly use this slide for your exam for good preparation. All the best, dear.